Hi, everyone. Welcome. My name is Noémie Elhadad. I'm chair of the Department of Biomedical Informatics at the Vagelos College of Physician and Surgeon, or Medical School. I'm also the director of the AI at VPNS initiative that attempts to promote AI across all of the missions of the school, which are research, clinical and care delivery, education and training, and community health. I'm very excited to welcome our speakers and our moderator for this session on cancer care from cells to systems. I think it fits very nicely after the previous session on brain and mental health, because you'll see a lot of the themes come back, and in particular this idea that AI now is pervasive from the basic science and discovery to translation into clinical care, and to shifting from, now that we have algorithms that are powerful enough to be accurate, how do we now think about the connection to humans and go from an algorithmic centric view to a human centric view of AI and really serving our patients? I am going to start the session with a technical talk from Dr. Raul Rabadan, and then we'll have a panel moderated by Dr. Gamze Gursoy. I want to introduce Dr. Rabadan. It's my pleasure to do so. Dr. Rabadan and I started as assistant professors many years ago. I won't say how many years ago, Raul, but it's been a thrill to see how much Dr. Rabadan has been able to do. Dr. Rabadan is a professor of systems biology and biomedical informatics at VPNS. His research is in computational genomics, and he leads the program for mathematical genomics at VPNS. He's at the forefront of innovation on multiple ways in which AI can be used in computational genomics, and in particular in cancer. Do you want to come, Raul, to the welcome?
Thank you very much, Noémie. We are in a very exciting moment, as you probably know. I mean, research has been changing, but this society as a whole has been changing through this AI revolution. I'm going to be talking about some of the work that we are doing here at Columbia by myself, but also mentioning some of the work in the program and by my colleagues here. It's very interesting. It's with applications in cancer, but it's many more things. I mean, with this idea of foundation models, we can getting a lot of data, like language models. We can get a lot of data, train these models in this amount of data, and then trying to different applications. The applications that I'm more interested in, because I'm working on this domain, is cancer, but is across many different biological disciplines. And one of the things that is happening that is quite interesting is biology has been always a science that is very descriptive. I mean, we get data from a patient, we can get single cell, we can get the genome, we can do clinical records, and we see what are the things there. And then based on this evidence, we try to decide what is the treatment or what's happening or what is the diagnosis. But one of the things that is changing is now we can start doing predictions. My background is in theoretical physics, and in physics, we have very good models for predictions. Once you have good models of how things work and you can do things, you can send missions to Pluto and arrive 10 years later, and they arrive there. They are not doing somewhere else. I mean, so I think here we can do something very powerful. I mean, once we have very powerful models. The first thing that we need for these models and these things is data. And as you probably know, we, in the beginning of this century, we started the human genome era. So the technology was there and the infrastructure was there. And then many people start sequencing the human genome, and then human genome becomes cheaper and cheaper. And then now we are sequencing many patients in cancer in particular. And people have been collecting here and many other institutions a lot of data from cancer genomes. So you probably know cancers, they are caused by mutations that we inherit from our parents. They are the germline mutations. These are contributing to risk. If you can, certain mutations can give you more risk of certain cancers. But most of the mutations that we are studying cancers, they are the somatic mutations, the ones that we get in our lifetime. If you smoke, you go to the sun, you inhale certain substances, all these things cause mutations, and you age also. Most of the mutations, they are related with age. So many people across many different institutions here in the US and across the world, they have been sequencing patients. And we have now probably close to 1 million patients that have been already sequenced. And then people are doing these cartographies. I mean, getting all these, you have the patient, you look at the mutations. These are somatic mutations in a patient, in another patient, in another patient. And say for one few cases in pediatric cancers, it's almost impossible to find two patients with the same set of mutations. They're all kind of different sets. But there is a structure in this data. There is a lot of structure in this data. So now when we have this cartography, these maps of how these mutations happened, we have to interpret and to make sense to all this data and correlate with prognosis, response to therapies, and many other things that happened. One of the things that have been very interesting is in the dynamics. I mean, cancer is our own cells getting mutations, and these are dynamic processes. Mutations, they occur at some point, and the cells start proliferating. There are different clones, different therapies select different clones, and these are very dynamic processes. And longitudinal studies, they are very informative of what is important and what is not. But one of the crucial challenges here is that we don't understand very well what most of these mutations are doing or what is the biological data that is there. So what we have now in biology, we have very, very good technology. We have high precision observations with mutations. We can see if a mutation is there or not. What is the fraction of cells that they have these mutations? We can identify using traditional machine learning patterns in this data. We can shoot some descriptions, and if you see, for example, single cell data, you see all these maps that happen with single cells and all these things. And we can start doing models, okay? We don't know what is the final methods that they are going to be kind of illuminating or predicting what is the best response. In my background in physics, for example, when Newton discovered kind of the Newton laws, he has to develop calculus, and it was taking like a century to make calculus kind of be a solid mathematical tool that can be used for solving differential equations and predicting different things. Here with AI, we are kind of looking at what is the right model. And what is fascinating about the field is that we have many different people coming with different models. Foundation models is one of those, and it's probably going to be the beginning. But we don't know if this is going to be the final model or not. From the theoretical point of view, we don't know very well how these foundation models or in general deep learning works. I mean, why is learning? Why these conditions? Why these structures? We don't know many of these things. So there are theoretical innovations that are coming along the way. I mean, better models, cheaper models, and more structured models. And the idea is to find fundamental principles that can organize all this information. So coming from the very beginning, from generating high throughput data, we are there extracting some of the data. We are just generating some of these models and then doing predictions for the future. And we have several models here. Some of the work here at Columbia by my colleague here, Mohammed Al-Quraishi, looking at the structures in different proteins, predicting very much like AlphaFold, predicting mutations, specific mutations, what are they doing in protein structures, of some of the work that we are doing and looking at non-coding mutations in cancers. I think one of the key ideas is this idea of generalizability. We have a lot of data. We can look at patterns in data, but we can extract some knowledge from these patterns and go in something else, a patient that you've never seen before, some structures that you've never seen before. The applications in cancer and beyond, they're manifold. I mean, they're from DNA, predicting things, some of the work that I was mentioning by Mohammed and myself, and both from academics and from industry. There is this crazy competition between different companies and different institutions trying to create better models, trying to predict things. From DNA, proteins, the structure of RNA and single cell, from molecules and chemicals, and to clinical data and pathology data. This is doing very good models. This is one of the examples that was the Nobel Prize last year, that you can predict accurately the structures of proteins, and then you can start doing perturbations on all these structures and trying to see how these mutations that we find in cancer can be affecting that. So here. So in the last few years, with colleagues here at Columbia and beyond, we've been mapping, doing sequencing many of these tumors and trying to understand what is the structure of all these tumors. I mean, some of the work we've been doing in brain cancers, in leukemias and lymphomas, and all this information now we're structuring, generating these foundation models based on a lot of single cell data, trying to see how the different transcription factors in different specific cell types, they are kind of associated to different programs in different cell types. And then we can introduce the mutations that we found in patients in silico. We can try to see what is the experiment doing experiments in silico to probe what is the biology of these things. We have a whole program here at Columbia, where we are just generating these foundation models and then applying it to many different things: brain tumors, DNA repair genes, pan-cancer studies, MDS, AML, stem cells, prostate cancers, and lymphomas, where there are different groups, both experimental, clinical groups, working on trying to understand what is the biology that is underlying these diseases using these foundation models. Summary, we're in a very, very exciting moment. We're in cancer research and anything else in the clinical side. Large data is enabling new models. The structure is there. We can do beyond descriptive to do something that is predictive. And models are very quantitative. When we are predicting the effect of expression, it's not only expression is going up or down, we can predict exact values of things that are going there. This is in cancer research. There are tons of opportunities here. I mean, we can generate these models, can guide experimentation, including experiments in silico. We look at the patient, we take the data, and we introduce that in our system and trying to see what's going on. It's allowing scaling and throughput beyond current limits. At the moment, if you want to do CRISPR, you have to do one mutation, another mutation. Here, we can introduce some of the mutations simultaneously in these genomes. It helps to integrate and collect and connect data modalities. I mean, many of us, we are collecting and working in many different aspects of many diseases. And what we are learning is just collecting all this information. We can learn from some of the data in one part of our institution to about something that is happening in a different part of the institution and all around the world. And I think the next phase also that is quite interesting is going to the generative part and is designing new promoters, genes, and proteins that can be doing specific things in specific patients. What we need, and I think Colombia is poised to have these things, we need multidisciplinary teams of quantitative and experimental teams and clinical teams, all of us kind of addressing common problems with different modalities and different ways of thinking. And the other thing as an academic institution is probably this revolution is not coming from us, of some of our colleagues here. We are going to be kind of the older people. It's going to be the new generation of researchers that are coming with new ideas, new methods. And our role here is to guide them, to form and guide all this new generation of scientists. As an academic institution, a research institution, I think we are kind of the best to do this kind of work that is going to be the leading work in cancer research and clinical care in the future. Thank you very much, and this is part of my team here.
Thank you so much, Dr. Rabadan. I just want to highlight the fact that something that Dr. Janet Wing said when she started the session, which is the short time it takes for AI fundamental research to make it into other fields, right? And so if we think about the first foundation models and large language models that came from text, we can be purist and say it started in 2017, 2023 is when the world is like, oh, there's such a thing as a large language model. And now we're in 2025 and we have researchers like Raul who are able to create foundation models of the language of biology, which is highly innovative. So thank you for that. So we're going to move on to the panel part of this session. And it's my pleasure to introduce Dr. Gamze Gursoy, who is Assistant Professor of Biomedical Informatics at VPNS and at the New York Genome Center.
I would like to invite all our panelists. Thank you, Noémie, and thank you, Raul, for the great presentation. And before I start shooting my questions, I'm going to first very briefly introduce our panelists. I'm not going to do justice because we have really highly accomplished researchers here, but I'm going to very quickly do that. And to my left, Dr. Chin Her is a professor of medicine and professor of epidemiology, a recognized expert in cancer screening and prevention for gastrointestinal cancers, and is a leader within a National Cancer Institute consortium of researchers that work towards improving cancer management. And to his left is Dr. Rita Kukafka, who is my colleague at Biomedical Informatics, who is a professor and also with a joint appointment in the Department of Socio-Medical Sciences at the Mailman School of Public Health, and she's leading a research program at the intersection of population health, data science, predictive analytics, decision science, behavioral science, and genomics. And I'm going to skip Raul, you already heard. So the next is Dr. Despina Kontos, who is a professor of radiological sciences and of biomedical informatics, with expertise in AI, machine learning, and big data analytics for multimodal imaging data. And last but not the least, Dr. Anil Rustgi, is the Herbert and Florence Irving Director of the Herbert Irving Comprehensive Cancer Center at New York Presbyterian and Columbia University, Irving Medical Center, and a world-renowned leader in the field of gastrointestinal oncology. And all of our bosses here, I guess, we are all part of Cancer Center. So with that, I'm going to actually start my question to Dr. Rustgi. As the director of the Herbert Irving Comprehensive Cancer Center, so could you give us an overview of the main areas of research and patient care at our cancer center in general and in cancer care, as well as within those areas, what role the data and AI currently play?
Sure. Thank you. By way of a brief background, our Cancer Center is a matrix cancer center that extends across Columbia University and involving 7 or 8 schools, although the crux of the membership and participation is at the medical center. There's over 200 members, 400 trainee members, and we're divided into 4 programs that basically span the continuum from very fundamental discovery science and cancer genomics and epigenomics, and Dr. Rabadan's co-leader of that program, and working with Dr. Al-Karoushi on, as you heard, on computational protein structure and function. So that's a point of emphasis in our cancer center and emphasis as well. And then there's another program, Precision Oncology and Systems Biology, that's co-led in part by Dr. Peter Sims of Systems Biology. There, the tools of AI are being more employed in terms of investigator-initiated clinical trials. So coaxing discovery science in the preclinical domain into the possibility of application in phase 1 clinical trials. So we put a lot of emphasis on investigator-initiated trials, and we're seeing enhancement of use of AI in that context. And then I would say we have a clinical informatics unit that involves Dr. Kontos, so she'll speak much more about that. That really involves the use of AI in 2 large areas. One is imaging, led by her in the Cancer Center, and again, she'll elaborate. But the other is in fundamental aspects of clinical care and electronic health records. Can we reach a state whereby if you're the provider and the patient is in front of you with various diseases or cancer states, can you bring up algorithms for management, what we also call molecular matching? If they have a certain mutational profile, can we match them with AI tools to the available clinical trials? So really, it's incorporating AI from discovery science to translational medicine to fundamental aspects of clinical care. And finally, we have a program in cancer population science. Can we then take these applications of AI into our population writ large and have an impact on cancer screening and cancer surveillance before we get to the stage of cancer therapy?
Thank you so much for this great overview. And now we can actually dive into the specifics, because I think we have representation from all of that you touched on. And I want to start with Dr. Her here. I was wondering if you could share how you use AI in your work, especially for cancer screening and prevention for gastrointestinal cancer. What specific research questions that AI is helping you answer, and what new possibilities that you see in the future?
Yeah, so cancer is very difficult. I think in some ways it's the most difficult disease area, and so hopefully AI will have the most to contribute. That's the big picture. So I'm a practicing gastroenterologist and a physician scientist, but I do mainly cancer population. So I started from an epi-type regression modeling type of machine learning, but then as my bread and butter was we built models, simulation models of cancer in the US population. So we would calibrate. So I developed a lot of understanding of models, not quite as smart as Raul, but we do something similar. We build natural history models of cancers and reproduce the US population, and then test different strategies, including screening strategies. What age should you start? How frequently for the different types of cancers? I've done that in esophagus, stomach, pancreas, colon, and liver. Those are the GI cancers. So the goal is early detection and prevention, right? As Dr. Rustgi said. So how can AI help in that. Right now, we have risk prediction models. Some of it, there's a difference between early detection. You find the breast cancer lump earlier versus metastatic. So that's important because there's survival benefit. But then in a lot of GI cancers, for example, colon cancer, we want to detect the colonic polyp, the precursor, before it turns into cancer, and then therefore it's prevention. So I'm a little biased. Colonoscopy is the way to go. So that's a little different, like trying to train a prediction model to detect earlier versus someone that will progress. And the latter is more difficult. You need observational data, you need time series data, and then that's where perhaps unknown risk factors. So most of you are probably familiar with, you can do a multiple variant regression using a logistic regression to see who progresses versus not. But we want to find hidden or unknown risk factors, and some of it ends up being the black box. But that's the immediate application. So anyway, that's the quick question. I work with someone. I don't think if Chihum Park is with me, but she's the real data scientist. I'm just the crazy MD. But
Thank you. So speaking of screening and prevention, early detection and prevention, so I want to ask Dr. Kontos, how would we do this from imaging and the modern AI? How does it help you to do what you do?
Great question. And I'll draw some examples from my own research, and then I can extrapolate a little bit further. So I'm a computer scientist by training and have been doing AI almost 20 years now, way before it was such a hot thing, I guess, and been living in a radiology department my entire academic career because I love working with imaging data. So in the screening setting, for example, there's a lot of information in imaging data that is not necessarily utilized by the current clinical evaluation. I'll give an example. I see many women in the room. So when you go and get your annual mammogram, the purpose of getting your mammogram has been so far mostly to tell if there is a cancer there or not, to detect. Research for more than 30 years now has been showing that mammographic density, and you may have heard this in the news as well because there's been a lot of discussion about it, is a risk factor for breast cancer. So the dense tissue in the breast, which is the fibroglandular tissue in the breast, as shown in conventional X-ray mammography, can predict which women are more likely to get breast cancer. And it can also tell us for which women mammography doesn't work very well because their breast is so complex in terms of the appearance of the tissue that it's very easy to hide, for cancers to hide underneath the complexity of the tissue, and maybe different modalities that may be more expensive or time consuming, such as breast MRI, need to be used. There's a lot of discussion in the field right now about breast screening, how it should be used, what frequency, which women should be getting conventional mammography, which women should add MRI, which women should get ultrasound, at what age should we start. No clear guidelines, really, if you look at it. And there's a lot of implications, both about finding cancers earlier versus later that are much more treatable, and also cost effectiveness implications. And MRIs orders of magnitude more expensive than a conventional X-ray, so we can't give it to every woman. It's not also available as broadly as conventional X-ray, and so there's all of these kind of complex decisions that need to be made. And this is a perfect example where AI can be used, for example. And this is an area of my own research for many years now, in different settings. How to measure, for example, this kind of breast density, the amount of the dense tissue. If you see how that is done clinically, is literally by visual assessment. And there are many studies that shown that if you put like 4 radiologists, they're going to assess your breast density differently. So whether or not there's going to be a decision that you're a high-risk woman for breast cancer and you need to get MRI may not really depend on your actual amount of tissue, but who's reading the mammogram. So there's a lot of subjectivity there. So AI can take that out of the equation. We can quantify, we can measure it very accurately, we can run the same mammogram 100 times, get the exact same value. So then we have quantitative values. That's great. But we can also use AI to combine different types of data that relate to the risk of developing breast cancer so we can have more comprehensive assessment of risk and more accurate. So we can put density, we can put genetics, right? We can put electronic health record data, we can put family history, we can put all kinds of information. And AI is really good at looking at the data and extracting patterns that you cannot see just by just conventional assessment or by human vision, and also mathematically combining the different sources of data in an optimal way. And so now we can start thinking about really personalized screening and precision screening. AI can help us derive an algorithm that can take with good accuracy which women are at high risk of developing breast cancer, which women need to get more intense screening, more frequent screening with additional modalities, but also look at the other spectrum, which women may be at lower risk of breast cancer. Maybe we also be thinking about de-intensifying screening for some women, because X-ray, may have, we have the radiation dose there to consider. Is not as harmful, really, as one mammogram. You get the same radiation as flying to East Coast to West Coast, so it's not, it's not that much. Still something to consider. So that's a very practical example of what we can do with AI, and in terms of cancer screening, for example, which I think is a great area of application. We can think about lung screening the same way, or other areas that we can leverage these technology.
Yeah, thank you so much. So while we are at the breast cancer, I want to zoom out a little bit and ask a question to Dr. Kukafka, because your research is on how technology can help patients and providers on their own and in their work together. And a lot of your expertise is in developing these technologies in cancer prevention. So as we develop better and better AI systems for cancer risk prediction, early detection, and care, we as a scientific community are finding out that it is not the end of the road. Now the providers and the patients need to handle this information and act upon it. And in the last session, we touched upon this a little bit. Can you describe the research challenges your work tackles and maybe some of the ethical questions that comes out of it?
Sure. No, now it's on. Thank you very much for that question. And so my work is basically leverages AI, standards for interoperability, and a patient-centered design to support patients in personalized decision-making and fostering meaningful communication between patients and their providers. We use, it's a data-driven approach. We use algorithms. So far, we've incorporated algorithms for breast cancer risk prediction into tools. And I have to say that we've been working on developing these tools probably for close to a decade, and have run several randomized controlled trials to evaluate outcomes of how these tools, when implemented into clinical care, actually improves both patient-reported outcomes and clinical outcomes. I think the high-level umbrella for thinking about the work that we do in my lab is in the space of precision prevention. So we really try to accumulate the data that we need to run these prediction models. And we move from a one-size-fits-all guidelines implementation, for example, for breast cancer yearly mammography, to a much more personalized, patient-centered approach to delivering risk-stratified or risk-aligned strategies to reduce risk. And the tools support both patients and providers. On the patient side, it is decision support as compared to education, because we spend time enabling patients to deliberate decisions that they have. We've done RCTs around genetic testing, chemo prevention. We just received 2 additional grants where we're going to incorporate both cardiovascular disease and breast cancer, 2 leading causes of death in women, simultaneously, to see how that impacts decisions. I do want to focus on a couple of the components that I think are important, and actually feedback to the panel, to the previous panel. And part of that is the black box. So what we do is the data we use to run these models include both data from electronic health records, but we also integrate patient-derived data. And the reason, as many of you know, is that electronic health record data is often missing data. There's sometimes errors in that data. So prior to, there's two purposes for integrating the patient data and the EHR data. One is for a more complete data set. But the other part is that patients are able to download their data using, someone mentioned the Century 21 Care Act, which requires that patients can access. So through our tool, which is called Real Risk, they access their own EHR data. They look at that data. They correct the data as needed. They add missing data, for example, family history, ethnicity might be missing. And then they actually see the data that goes into the risk prediction models. Once we, and that requires both the algorithms and also the standards for interoperability. Someone also mentioned at the last panel that communicating that risk is important. We want patients to make informed decisions, so we spend a lot of time really thinking about and evaluate different ways to communicate risk. And all of this, I should say, is outside of the clinical encounter. So one of the things that we do in this decision aids is they have interactive games where we rig pictographs so they could see what a 12% or a 20% risk looks like. So we do spend time, we do the same kind of gamification to talk about risk and benefits of different risk-aligned preventive options. So they understand their risk, they understand what they can do to reduce that risk or impact their risk, and then they're encouraged to take, they have an action plan, they're encouraged to take that to their provider to discuss during an encounter. Providers also have decision support, which I won't talk too much about, but the data is communicated both to patients and providers. So that's pretty much, and summarized in 5 minutes, but it's been 10 or 15 years of work and a lot of funding to get here. But I think that the part that I think is most unique is that we have figured out how to leverage AI interoperability standards for data integration. So we are able to collect the data not just from the clinical data, but also patient data that they can best contribute. And we promote this through a patient-centered design. And I do think that will bring us to the next phase of how do we actually move AI into action so that it improves, so that patients can actually look at these predictive models and it becomes actionable both for the patient and for the provider.
Thank you so much. And now, while we are at AI in action, I want to now zoom into the cells and ask Dr. Rabadan a question. So I'm sure there are a lot of cancer biologists who got very excited reading your paper, but they probably don't know how that can help to their research. So can you tell us what they can do with these foundation models? Can they design the next experiment or hypotheses, or what kind of downstream tasks that people who are not developing the models but using the models can do?
Yeah, there are many, many different directions. One of the things that is quite interesting being here at Columbia is that we have very interdisciplinary teams of people. So, for example, I can mention we are working with some groups working in B-cell lymphomas, T-cell lymphomas, different groups. They have their own interest. Some of those they're interested in, for example, trying to understand what is the impact of some mutations in non-coding parts of the genome. They publish some work, but they are been mapping some of these mutations. Most of those, we don't know what they are doing. So we are co-developing some of these methods with one application in mind. The models, they are kind of huge. I mean, so you can learn many things with having some application in mind, also guide the development of these models. So we are working with these teams, optimizing the models, applying it, developing some of the statistical framework for thinking about the impact of some of these mutations. Other groups, they are working in the microenvironment, for example, trying to see how the other cells in the tumor, they are communicating with cancer cells, promoting or just the immune system, how the immune system is working with them. So all these different applications, there are different ways that these models can evolve. It's very hard to develop a model that works for everything at the moment, but they are kind of working with different teams. We have different applications in mind. And that's the way that we are at the moment. We are just following, just working with our colleagues here, looking for funding, looking for data, and generating data, and designing experiments all together. So I think the collective endeavor here is quite important.
Thank you so much. So now I'm going to ask open-ended questions, and that might be a little annoying for the panelists. But first, I want to ask to our clinician scientists, and I want to ask, where do you see AI in 10 years in terms of clinical care, and then feeding back to the research that you're doing, Dr. Rustgi and Dr. Hoover?
Anil, Dr. Rustgi, would you like to? Okay, so that's really difficult to answer. So, but I'll try. So a lot of what I started doing a while ago in terms of risk prediction, risk stratification, that used deep learning or neural networks, right? So I was trying to identify hidden risk factors or things that were not obvious. But as we all know, ChatGPT, et cetera, the generative AI, and it's interesting. How are we using it now in the work that I do? So, for example, they're very good at cleaning data, because especially if you're doing EHR data, that's the hardest part. When I first hired my person, I said, I want you to download all the Columbia pancreatic cancer cases and just start cleaning, because we're going to mine the heck out of it. Right? We're going to do keyword searches and then correlate it with lab data, time series data, and predict someone who's going to develop pancreatic cancer. 4 years later, we're still trying to clean the data. It's very, and there's thousands and cases. So that's going to be big. I know that sounds like low-hanging fruit, cleaning data. I mean, genomics and radiomics are a little better because ultimately they're ones and zeros. So we homed in on laboratory data, for example. So I think that's gonna be big. We have used it also in the GANs generative adversarial network. We've created phantom images, particularly when there weren't enough images. But it's a little tricky when you don't have enough data. You don't have enough data. So we're trying to. I think in engineering this is used more frequently, but we're trying to create synthetic data, particularly of patient phenotypes that aren't as common. And then we can run simulated clinical trials. I focus on screening, but that could also be applied for treatment. In terms of decision support, everyone knows, you're in your Epic thing and beep, beep, something pops up. This person's going to develop pancreatic cancer, order an MRI now. Yeah, I think so. I think a lot of people, that's what they think of, digital health. I'm not as much in that space. I think, a lot of times when the current risk factors we have are sometimes pretty good. So it's more about pushing the boundary, finding it earlier. And we need to be careful. I mean, ultimately, like computational people, if you have enough cases, eventually central limit theorem, you'll be able to predict everything perfectly, right? But it's going to be a long time, especially with cancer. It's so complicated. You have genomic, you have genes that you're born with, but the environmental exposures are very, very complicated. And it's not like asthma, high pollen count, you have a flare. These environmental exposures could have been 40, 60 years ago. This is going to take a very long time. So I think we need to at least start until we have perfect knowledge. I think we need to start with the low-hanging fruit. Like, for example, the example I'll give is I'm quite interested in liquid biopsy for cancer blood tests, for cancer detection. It's a very hot topic. And we need to be careful. There's the first commercial product screens for 50 cancers simultaneously. And we need to be careful because every cancer is different. And we can do a lot of harm when you say someone's positive. You're not sure if it's a false positive, and all the difficult situation it creates. So we need to be very careful. For example, going back to colon cancer, if you have a positive test and it's a false positive, we do a colonoscopy. And then we can assure the patient that they do not have colon cancer. I mean, nothing's 100%, but we can look them in the eye and say, you don't have colon cancer. Whereas in other cancers, it's so we need to be specific. Like just like you've seen one cancer, you've seen one, you need to be careful about the clinical applications per cancer. I know Raul's going the other way with foundational models, and that's more for biology, but yeah, so that's a long answer.
I think a theme that you're hearing on a recurrent basis is cancer prevention through screening. And there are many meritorious aspects of that pursuit. Right now, healthcare in the United States constitutes about 24%, 25% of the total GNP. And yet, it's not regarded as the absolute best healthcare system in the world. Our maternal mortality rates are high. Our fetal rates are high. All sorts of problems with aging. And I think a bold vision requires intervention using AI tools, as it is increasingly in the consumer world, in the healthcare world, and that will lead to an impact that will be positive on the national economy. So I think we've got to marry AI with very practical healthcare considerations at a societal level.
Thank you so much. So now we're talking about the 10 years vision of AI in cancer. I want to ask Dr. Rabadan a question. So 10 years ago, when I was a young grad student, when they told me that, let's do protein structure prediction. And I could never imagine at the time that AlphaFold is going to be a thing. And I said, this is such a boring problem. Everyone is working on it, and I don't want to work on it. So then I ended up working at ENCODE Consortium, where we were trying to basically functionally annotate all the non-coding mutations. And again, I saw that as maybe not a problem that we can solve. So do you think that in 10 years, with the help of AI, are we going to understand the functional role of every non-coding mutation in our genome?
I don't know, but I mean, I think we are getting closer to that. I think it's clearly, I mean, there is a lot of information encoding in, let's say, for example, something that is happening in a cell. I mean, all cells, most of the cells, they have the same DNA. They express different genes, and there is information in every cell, and different cells, they have different parts of information that is informing other ones. Other organisms, mouse and humans, I mean, from the point of view of cells, they are not so different. I mean, many things there. So we can leverage some of this information. There are many models organisms, even a fly or a C. elegans. I mean, some of the biology is the same. So if we can start incorporating all this information, we can get very rich annotation and functional annotation of some of the role of some non-coding parts in the genome. So I think in the future we get closer. I mean, that's for sure. I mean, how close to the final response, we don't know, but I think closer. And as we are getting richer and richer, I mean, the model that I was presenting is only human, but the team now is working on mice, creating one for mouse. And perhaps just getting more information about different species is going to enrich our knowledge of the biology and different conditions. I mean, cancer is a particular condition, but you say that you have an infection, for example. Cells in an infected condition, they are very, very different from the other ones. I mean, so once we get more information, we can start learning some of the structure that is there. And different people and different communities and different ethnicities, I think is quite important. So the more diverse information, the more we can learn about all these parts of the non-coding genomes.
Thank you. And Dr. Kontos, where do you see in 10 years the radiology and AI in the same way?
So first of all, to say something about radiology and AI, I didn't even know this, but 80% of the FDA cleared AI tools are on radiology applications, which is great, right? Now, as a computer scientist who lives in radiology, one of the reasons that I chose to live on the medical side of the spectrum is because I would ultimately like to see some of the tools that we make in a laboratory setting make it to the clinic and be something beyond just a publication or something that inspires, the development of tools that actually make it to the clinic. And so I would like to see a more rigorous process of evaluating the algorithms we create, the tools we create, in terms of putting them in the clinic, a more scientifically kind of established path for doing that, but at the same time, a more streamlined way of doing it. So that, right now we're not good in either. We don't have a very rigorous way of evaluating these tools in general, and not just like the academics, like everybody. And we also don't have a very well streamlined way. It's amazing. So when I was a grad student, I decided to go to medical school for my career because I saw radiologists doing the resist measurements by hand. If you don't know what a resist measurement is longest tumor. So in treatment for cancer, an indication of whether or not a patient is responding or not is to estimate the longest diameter of the tumor. And if it's shrinking, it's working. If it's not working. So the way they were doing that would literally take the mouse on the screen and click on the MRI on the workstation visually. So if I'm doing it versus you doing it, could be treatment's working, treatment's not working. It's kind of still happening the same way. And we have, I don't even know how many, I don't know, thousands of tumor segmentation methods published in the literature, right? I would love to see that gap close as much as possible. And I do think this is an area that academia has a strong role to play in creating more creative partnerships with industry. I am very passionate about that, to finally see some of these things make a difference.
Thank you so much. And along the same lines, Dr. Kukafka, as we close this gap, how are we going to interact with the patients about AI in 10 years? What is your vision?
So my vision is a prediction that might seem out of maybe off the wall, but I think that there is potential, and I think it might happen, that AI will be, and it's happening now, I mean, direct to consumer AI, to the point where as healthcare organizations become more difficult to navigate, as primary care providers are less and less, as patients have more difficulty accessing healthcare, and we see this now that these ChatGPT, large language models are, we see startups promoting AI directly to patients for diagnosis, for treatment. And I do think we're going to see more and more of that. In part, I mean, there's many logistical reasons, not the least is the amount of capital that industry has to test different applications, which becomes more difficult in academic settings. That we see that some of these industries incorporate providers so they could prescribe, they could treat. So I do think that as our perspective is largely from an academic and healthcare organization perspective, the world is moving, and it's moving outside of healthcare organizations. And I think AI and access direct to patients is going to be commonplace in 10 years.
Can I just add something? I think the reason it's so difficult, because you said 10 years and I said, no, that's hard. I think it's, as we all know from the press and whatever, the generative AI, it's growing so exponentially. We don't know what ChatGPT 5.0 is going to look like, but it's growing so rapidly. And we don't even know how to conceive of how it may help us. I think so. This is a good thing. I mean, we don't know. I mean, we haven't even figured out how it might help us, but especially when they have better inference. Right now, they're training. It's, but I think the one of the obvious things is cancer drugs, and this is more a Raul space. But I think it should happen. I can't conceive of exactly how, Raul. I don't know if you can, but anyway, I want to. But this is an exciting time. I think this is a really exciting time because, I'm used to thinking along deep learning and we can find more hidden data. But this is another, once we get inference, what is possible, we don't know.
But as a physician, if you were to pick a problem that is not unsolved and ask computer science community to develop something, what would be that problem in cancer care? Yeah.
Well, I'm a prevention guy, but I think drugs would be fantastic. If you can cure cancer, and whether it be drugs or genetic modification, whatever. And I think that's where a lot of precision oncology is. We're trying to understand pathways so that we can design drugs that are specific to the pathways and mechanisms. But and these are very complex systems. As Raul's a pretty smart guy. These are very, very complex systems, and I think we'll have a lot of elucidation of this in a highly accelerated rate. Raul, I mean, I'm speaking out of turn, so I mean, tell me if you disagree with some of what I'm saying.
No.
No, like you agree, or no, like I just don't know what I'm talking about.
No, I think we don't understand very well what is the future. I mean, it's very hard to predict. Yeah.
So while I have you, Raul, so there are a lot of virtual cell initiatives now, in the field where people are trying to build a virtual cell using foundation models. Do you have any of that kind of work in your lab, or what do you think about those?
No, I'm not involved. I know that Mohammed, the person I was mentioning, he was in one of the papers. I'm not involved. I'm just looking at only specific, some particular 2 domains or 3 domains, so genetics, epigenetics, and transcription at the moment. Doing something much more ambitious, I think, requires a big team. I mean, it's probably going to be like kind of a Manhattan project of many different people going from modeling protein structures. I mean, organization, cellular organization is going to be very complex. It requires kind of an army of people. I have a much more modest.
I want to ask any last thoughts on the future of AI from any of you, or the AI at Columbia in cancer research, and what do you want to see, or what are the things that are exciting you right now? Again, another open-ended.
So something that's exciting and a little scary. I think foundation models might put a lot of PhD theses out of work. Right? We used to have like theses around, for example, in my field, medical image registration, medical image segmentation, whatever. And all these things are going to be simplified, much easier done, which I think leaves us with answering the more interesting questions using AI, right? So there's a lot, in AI for doing some basic tasks. We're going to see a lot of that, which is great. I think that's going to give us the freedom to think more creatively around asking interesting questions.
Yeah, I want to mention something that I mentioned in my presentation. I think we are in a very exciting moment and a very exciting place. I mean, here we are developing these tools where people from computer science, we have a great computer science department, people in math, applied math, we have people doing basic biology, people doing clinical work. We're in a big institution with many different expertise. And developing a tool is not something just developing a tool itself. I mean, it's in the context of different applications. And I think an academic institution with all these different domain expertise is the right place to be. And as I was mentioning, we don't know if these foundation models that we have right at the moment, they are going to be the right tools. This is evolving very fast. We don't understand the foundations of these things. Why this architecture, not another architecture? Why these parameters and not other parameters? It's driven, and this is very exciting by practitioners. I mean, the theory is basically absent. So from people doing some of basic foundation of these foundation models or these deep neural networks, doing something from the basic for the applications and having a university with this portfolio of intellect, doing all these different things. I mean, having the right teams here is what is going to drive the field. And it's probably not being myself or others, it's going to be the new generation. So I think it's very exciting for the new people coming in the field because they are coming with new ideas, new methods that we are not even thinking about. And combining the expertise of someone working in clinical care, or someone in imaging, or doing what we're doing or what you are doing. And I think now it's kind of a wild field, and I think it's a very, very exciting moment. It's a revolution, and this is the right moment to be here. It's just in the middle of the mess of the revolution.
Well, thank you so much, all of you, and thanks everybody